some have the short span of life. How does that happen?
Well, could you just repeat the first question again?
The first question is how do immune boosters differ from antiretrovirals?
You mean, how does immune treatment or immune response?
There are these things that are being sold here, mostly in South Africa. They call them immune boosters. Every shop you go to,
Immune
boosters.
Oh, boosters.
Yeah,
Oh.
differ from antiretroviral.
Okay, so as far as the first question is concerned, since I don't know what these immune boosters consist of, I can't give you a specific answer. But what I would tell you is that now that I think a lot of you have more of a context of the immune response, you can appreciate that immune boosters may be a double-edged sword, because what HIV preferentially infects are activated T cells and macrophages. So if you have a resting T cell, it's a lot more difficult to infect it with HIV. People who work in the laboratory know that if you take peripheral blood from a patient, put it into tissue culture, and add HIV, it's very difficult to infect. The only way that you could really infect those cells is that you need to give them in vitro a mitogen, interleukin 2, maybe anti-CD3 and anti-CD28, and then only then can get infection, right? People familiar with that? Well, think in terms of a patient. Immune boosters may actually be, again, if they make the T cells activated, they may actually make things worse. And in addition, people who have an underlying infection may be more susceptible to HIV because their immune system is activated. So again, I just want to, number one, say that if it really does activate T cells, it may actually not be beneficial. But the second point I would make is that antiretroviral therapy works. There's absolutely no question about that. I mean, anybody who has taken care of patients before ART was available knows what the natural course of the disease was. Within a period of time, patients went deteriorated, ultimately died and lost weight. With the whole clinical spectrum, when you put patients on antiretrovirals, they can live relatively normal lives. So ART definitely works. Okay, so immune boosters, I can't give you a specific answer on. I mean, clearly, and again, it depends on, and that actually goes to your third question. The third question is, why do some people have a more rapid deterioration than other individuals? And again, that's one of the reasons why a lot of the studies are going on here, looking at a large group of patients to try to start answering those questions. But one observation has been made clearly that HLA type plays a significant role. So for example, HLA-B57 has been associated with individuals that are able to control HIV infection better than others. Well, why should HLA play a role? Well, now that you've learned some, immunology, what would you postulate? Why would someone who's HLA-B57 be better able to control HIV infection? Well, okay, let's think about it. What do MHC molecules do? They present peptides. And what defines what kind of peptides an HLA molecule can present? The anchor motifs, right? So again, I'm just throwing this out, but it may very well be that some HLA types may have unique anchor motifs that uniquely allow them to express peptides from HIV that may not be that may be from highly conserved regions that are more difficult to mutate. In that case, if you mount a strong CTL response against those epitopes, it may be able to control it better. I mean, that's one possibility. But again, clearly HLA is variable. And again, we all know that everybody's immune system is different. So there are some people that they get a cold, they can't clear it, they're always coughing, and they're always getting one infection after another. And other people are never sick a day in their life. And part of that, we say, is because the quality of people's immune responses may be different, and that translates into HIV. And the other answer that people have proposed is it depends upon the strain of the virus. And that's, again, another kind of semi-controversy. Is it the virus or is it the immune system? And when I was at Aaron Diamond, I think about 10 years ago, to give a talk, everybody there was wearing a button that said, "It's the virus, stupid," because, like, that's what they thought. It has nothing to do with the immune system. It has just something to do with the virus. And I'm sure that in Bruce's lab, people were walking around with buttons saying, "It's the immune system, stupid." And clearly, it's a combination of both. But there's no question that there are some strains of HIV, because of the fact that their long terminal repeat or their accessory proteins may work a little bit better, are more virulent. So that is another possible explanation of that. And the second question is, how does nevarapine prevent transmission? Again, it's actually quite straightforward. The lower the level of virus in the patient, the less virus there is to be transmitted. So if you treat somebody and you bring their viral loads down to non-detectable levels, there's just not a lot of virus in the bloodstream, so therefore, either at birth, when the baby's exposed to maternal blood, or potentially in utero, you just don't have that same level of exposure to the high level of inoculum. And the same thing is true in terms of transmission. Individuals that have high viral loads tend to be higher transmitters than individuals that have much lower viral loads. Okay, so I mean, classic. If you would, in the experiments we've done, if you would take, like, a million salmonella and drink it, you can get very sick. If you take like 10,000 or 1,000 salmonella, it doesn't bother you at all. So clearly the amount of the inoculum plays a role. And what the nevarapine does is it dramatically lowers the maternal level of HIV, and therefore when the baby is exposed to the blood, it just don't have that much virus there to infect the baby.
So does that mean that if you come into first contact with HIV, you don't get infected?
No, if you can absolutely be infected with HIV the first time that you're exposed to it. There's no, if it's a high enough inoculum, you can be exposed to it. You can get infected.
So it depends upon the virus titer in the body.
Well, that's one variable. I mean, the numbers that people quote is that the transmission rate in terms of sexual encounters is between 1 in 200 and 1 in 400. So that sounds, like good odds, right? But we all know that 1 in 200 could happen the first time, or it could happen the 200th time. So that it's all a question of now. If someone has 200 exposures, then the chance of them getting infected are pretty much going to be almost 100%, just from a probability point of view. But there's also variables in terms of mucosal tears, in terms of blood-blood exposure, so there's a lot of variations. And as you know, things like circumcision is another variable that people have identified in terms of transmission. Okay? Okay, fantastic. So again, for me, the most exciting part of teaching is actually stimulating questions, because when you stimulate questions, you're really demonstrating that, number one, people have an understanding of the material, and the second point is that people are thinking, and triggering thinking is really the most important thing to do with students, because as we get older, older people really aren't as good at thinking, so we need younger people to do the thinking for them. Question?
I have a question. In terms of genital mucosa, if you're looking at the vagina or cervix, the HIV will exploit the normal route with maybe Langerhans and dendritic cell into lymph nodes. But there seems to be some evidence of it entering into the epithelial cells with transcytosis. How do you explain that?
No, the way I would explain it is that there are multiple routes that the virus can take, and which one it chooses to take, it's maybe random, but at the end of the day, the virus ultimately has to come in contact with a cell that it could productively infect. And to my knowledge, there's not a lot of evidence that epithelial cells can be productively infected in a way that they then turn around and make a lot of virus. So even if you infect an epithelial cell, if the virus sits there integrated but doesn't make any more virus, then the patient's not going to get infected. So at the end of the day, the virus has to infect a CD4 positive T cell or macrophage in order now to trigger the cycle of expansion of virus and then expanding infection. But there's no question, there's more. It's like a building. Okay, most buildings have more than one entrance. So which entrance that you choose to use, it's a variable.
This is the route in terms of being in a vacuole as such. So even seems to be some talk about epithelial cells in the kidney, for example, that I'm aware of, that might be latent reservoirs. There's not enough evidence, but there's talk about it.
Right, and in the early 80s, there was talk of HIV infecting colonic epithelial cells. There was actually data for that, and it probably does. But again, in terms of a latent infection, you really don't need a lot of virus as long as it then is able to infect a T cell where it could then take off. So think about like a fire. If you have a tiny spark, but there's no material that that's flammable nearby, you could be making sparks for your whole life, and you're not going to start a fire. But if you do sparks right in front of very, very flammable tinder, one spark falls on that tinder, and then poof, the whole thing goes up in flames. And if it's in a house, the whole house can burn down. So in terms of being a reservoir for latent infection, you don't need to pump out a lot of virus as long as those virions can now turn around and infect a T cell or a macrophage that can now dramatically expand it. Okay, does that help? Okay, so any more questions? Yeah.
I've always assumed that trauma played an important role in sexual transmission of HIV. And because you needed some sort of break in the mucosa. But it appears that that might not be the case, because you mentioned that dendritic cells do have the ability to... So how would you explain that the increased transmission in homosexual behavior as opposed to heterosexual behavior?
That's a question that a lot of people have debated for a long period of time. There's no question that trauma facilitates the ability of HIV to get into the bloodstream and cause infection, get past the epithelial barrier and cause infection. And in fact, in animal models, if you traumatize the genital mucosa, you actually have increased infection rate. And clearly, there's also the issue of co-infection. So in addition, some individuals may be infected with other sexually transmitted diseases, which also compromises their mucosal integrity. So again, that is an addition. So you clearly can get transmission across an intact mucosa, but clearly if there are breaks in the mucosa or the mucosa is compromised, you probably increase the transmission rate, which makes sense.
And would you say that STIs increase that risk because you're more likely to have dendritic cells? Would dendritic cells come into the mucosa at a higher rate?
I mean, again, you're on clearly postulating things that may very well be the case. You have a lot more dendritic cells, they're activated. Your T cells that are present there are activated. Again, as I mentioned to you, as you know, HIV preferentially infects activated T cells. So if you have a STD going on and you have a lot of T cells that are activated in the lymph node, now within the dendritic cell brings the virus to the lymph node, that you probably have a higher chance of getting infected. Okay, great. Okay, so now this lecture is actually talking about failures in host defense mechanisms that are congenitally immune deficiencies that have a lot of the similarities to HIV, but they're not caused by an infectious agent, they're caused by mutations. And questions that I want to consider, and very few of you here are clinicians, so the way I like to structure this lecture is not like these are the immunodeficiencies that patients can get and you need to know about them, but rather what insights into the working of the immune system can we learn from patients? Because a lot of these patients have mutations that affect immune cells, immune molecules, and then the question is, what are the manifestations of that? And a lot of things that we rationally think should happen because of what we know about immunology from in vitro studies turns out not necessarily to be true when we look at patients. So the same way you do an experiment with mice, you knock out the gene, see what the phenotype is, to a degree, we can do the same thing in humans. They have genetically knocked out genes because of a mutation, and now we look at the patient, see what the phenotype is, and that teaches us what proteins and what signal and what pathways are critical in having an appropriate immune response, and then we could dissect exactly what stages these different mutations impact on it. How does, this obviously is not as relevant to you, but how does one elicit a clinical history? But it's also interesting just to know, how do we utilize our knowledge of the maturation of the immune system to understand what kind of pathogens can infect an individual? And, well, how can ignorance of immunology kill your patients? So if people are interested in learning about that, I'm more than happy to teach that. But This is a picture of David, and if you recall from the first lecture that I gave, which is only 4 days ago, but it may seem like a while, he had severe combined immunodeficiency, and when he was born, he was placed in a bubble, and this actually illustrates one of the early bubbles that he was placed in, and all the toys that he had, for example, everything had to be sterilized. So clearly this is before Fisher-Price, because all they had back then were blocks. And then here you could see this is a glove box. So the same way when you're doing PL3 TB work, everything is done in a glove box. That's the environment that he lived in. Because if he had no functioning B cells, T cells, and NK cells, he was basically susceptible to infections with every pathogen. So the way that he was being protected was by putting him in a sterile environment. And therefore, even though he had no functioning immune system, he wasn't exposed to any pathogens, and therefore he was able to do fine.
Did he have a sterile bowel?
The question is, did he have a sterile bowel? And they actually did some stool smears, and he probably had some commensals that weren't affecting him. It's another interesting aspect that if you have mice that are nomobiotic, they're completely sterile, a lot of them actually develop volvulus of their intestines because having no commensals actually is apparently not very good for it. So he may have had low levels, and they actually, the original papers probably they did some stool smears, but I don't recall that data. But I totally would say that he wasn't absolutely positively sterile. Now you also have to understand something else. He had a perfectly functioning innate immune system. His macrophages worked fine, his polymorphonucleoleucocytes worked fine. So in essence, it wasn't like he had no immune system. And as you learned from one of the lectures, if an angel asks you, what immune limb would you want to give up, right? You'd rather give up your acquired immune as opposed to innate. And he had a fully functioning innate. And that may be sufficient to control small inoculums of commensals. And as he grew up, again, he got larger and larger quarters. But again, here's another glove box, for example. And again, everything was sterile. And this is what he looked like when he was becoming a teenager. Again, everything was done through gloves. And again, as I had mentioned earlier, unfortunately, he passed away. But the question at that time was, why did he have an immunodeficiency? And at that time, no one could really answer that question. And in addition, what can we do to treat him? Now, as opposed to a patient with HIV where you could treat with drugs because you could basically prevent HIV replication and therefore now treat the underlying cause, and in the case of a congenital disease, that won't help. The only way you could really treat this individual is by correcting their genetic defect. Well, back then, the way they tried to do that was by giving a bone marrow transplant, by giving cells from someone that had a functioning immune system, but unfortunately, he succumbed to complications of the treatment. But as I'll go on, since then we now have developed technology that actually allows us to treat this disease, or this congenital disease. Now, if you ask the question, what are the causes of immunodeficiency in the general population, this is a very, very helpful pie chart, because over half of them are purely B-cell immunodeficiencies. And again, based on what you know about immunology, it probably makes a lot of sense, because immunoglobulin rearrangement is a very complicated process. It involves a large number of proteins, and there's a lot of places for things to go wrong. Phagocytic diseases are about 14%, and combination B and T cells are about 25% of the immunodeficiency. So again, the numbers all suggest the most common are B-cell immunodeficiencies. If you want to evaluate a child that has you suspect of congenital immunodeficiency, the first question you want to ask is, do they have increased frequency of infection? Now that makes a lot of sense, because if you don't have a good immune system, you get a lot of infections. But what's also important is the inability to clear infections. So normally, we get sick, we're sick, but then we get better because we're able to clear, we mount an appropriate immune response, and then we clear the infection. If you lack the ability to clear the infection, that suggests there's something wrong with your immune system. So you always have ear infections, you're always coughing. Also, what's a really red flag is infection with opportunistic pathogens. And that many of you are familiar with HIV. It's the same concept. Someone who's infected with pneumocystis, for example, it doesn't normally infect individuals with a functional immune system. That suggests that something is wrong, because normally these pathogens are very easily controlled by the immune system. If you are having infection, it means that there's something wrong with your immune system, that you can't control these. age of presentation is actually very, very important. So if patients are fine until 6 months of age, that indicates an antibody deficiency. Can anybody tell me why that should be? Excuse me? Exactly, maternal antibodies, because maternal antibodies cross the placenta in utero, and the half-life of IgG is between a month or two. For the first 6 months of life, the baby still has enough antibody to protect the baby. By 6 months, the antibody is gone, and then the individual is susceptible to infection. However, if they report being sick in the first few months of life, that now suggests they have a cellular immunodeficiency, something wrong with their T cells, because clearly maternal T cells do not cross the placenta. Okay, so that's right there, without even doing any lab tests, you could actually get a tremendous amount of information. And this is to illustrate the point that you made, is this is in utero, and during placental life, maternal antibody, specifically IgG, crosses the placenta, as I had mentioned previously, through active transport, through Fc-mediated transport across the placenta. And for the most part, IgM does not cross the placenta, IgA does not cross the placenta, only IgG does. And in fact, at birth, the baby's IgG levels are actually slightly higher than the mother's. Now, like a minor point is that if you have a premature baby, so let's say you have a baby that's born at 28 weeks, which is about 6 months, well, you look here and you see that the levels of IgG that have crossed the placenta is much, much, much lower. In fact, that's why premature babies are at much higher risk for developing infections, because they have not yet gotten that high level of antibody, which probably comes in the last 2 to 3 months of gestation. So that's something also that has clinical relevance. This particular slide actually shows an example, what's called transient hypogammaglobulinemia of the newborn. For some reason, some babies, takes them a little bit longer to initiate their production of immunoglobulin, but again, it's a benign condition, and just a matter of waiting for their immunoglobulin ultimately to kick in, and afterwards they do very, very fine. Okay. In addition, as I mentioned before, you want to look at what pathogens are infecting them. So again, based on what you now know, if an individual is getting a lot of bacterial infections, what do you think their defect is going to be? Antibodies, because that plays the critical role in protecting from bacterial infections. If they have a lot of viral infections or fungal infections, what limb of the immune system do you think is compromised? T cells, because that plays an important role there. So again, by getting a good clinical history and taking what you know about immunology, you could actually make a very good educated guess about what's going on. This is basically a review is showing that you have the pluripotent hematopoietic stem cell. It differentiates to the common lymphoid progenitor, B cell, T cell, and K cell, and on, and then the myeloid differentiation. But the point of this slide is, again, just to tell you that clearly different genes play critical roles at different maturation forks in this differentiation pathway, and if the mutation is at that protein, then you impact any of the downstream maturational processes. So now, is having a mutation that prevents maturation of the pluripotent hematopoietic stem cell, is that compatible with life? Why not? Well, right, you're not going to get platelets, and you're not going to get red blood cells, so we don't see that. Clearly, so for the most part, we don't see a lot of myeloid mutations in terms of differentiation. But again, if you have a mutation that affects the common lymphoid progenitor, you won't have any B cells, T cells, and K cells. If you have a mutation that affects T cell maturation, you won't have T cells, but you'll have normal numbers of B cells. And you could have, if you have a mutation that impacts on subclass immunoglobulin maturation, you'll have normal B cells and T cells and K cells, but you'll lack immunoglobulin subclasses. Okay, so that is pretty straightforward. And again, as you all know very well, you could basically characterize lymphocytes based on unique surface markers they express, and this is the way we would evaluate a child with suspected immunodeficiency. Ask, what are the cells that this individual has? And it's kind of like, I guess, in a soccer match, if you want to know the team is losing and you want to know what's the problem with the team, you have to look at the individual players and say what player is not playing well or what player is missing. So for example, if you just happen to forget to have a goalie on your team, right? You could imagine that the other team is going to score a lot of goals. So you basically analyze and say, oh, I'm missing a goalie, that's what the problem is. Clearly, that is a fixable problem. So this way you know, do you have B cells, do you have T cells, do you have CD4 cells, et cetera, et cetera. And again, all of you are familiar with this, basically using flow cytometric analysis. This is showing a two-color analysis, and this is looking at B cells. And here, this is IgM versus IgD, and here you have a population of cells that is both IgM positive and IgD positive, and what would you call this kind of cell? A what? A mature B cell, because you know enough now to know that if this B cell expressed IgM, so you'd have a very nice population here, but it did not express IgD, what would you call that B cell? You call immature B cell. So again, this is a very nice way of identifying both maturational state as well as the cell population. So what David had was severe combined immunodeficiency. He had markedly decreased T cells and B cells. Interesting that some of them actually could have normal cells. The cells just for some reason just aren't very functional. He had markedly decreased immunoglobulin level. It presented pretty much after birth. And these individuals can be infected with bacterial, viral, fungal pathogens. If they don't have functioning B cells and T cells, they're basically wide open to the spectrum of infection. The etiology is a mutation, and therapy at that point is bone marrow transplant, and I'll discuss a little bit later how gene therapy is being utilized to treat this. Now this is showing, again, the hematopoietic lineage, but in the case of David, where it was SCID, it was X-linked, which means that it affected males and did not affect females. And therefore the postulate is that there must be some mutation that is affecting a protein that plays a critical role in NK cell, T cell, and B cell maturation from the common lymphoid progenitor. Okay, that's clear? But what's interesting about David, for example, is that David otherwise was perfectly normal. A lot of the immunodeficiencies affect proteins that not only are critical for the immune system, but they're also critical for other aspects of differentiation. So they'll have other manifestations of the mutation. But in this case, it was purely impacting on the immune system, which led to a question of what gene is only affecting the immune system. And this turned out to be a really very interesting observation because, as you recall, the interleukin-2 receptor has an alpha chain, a beta chain, and a gamma chain. But then when they did knockouts of interleukin-2 in mice, it turned out the mice had, as you may recall, had a relatively normal immune function. If you knocked out the alpha chain and the beta chain of the interleukin-2 receptor, it turned out that they had a similar relatively normal immune system. But then they knocked out the gamma chain of the interleukin-2 receptor, and guess what? These mice had severe combined immunodeficiency. So intuitively, it made no sense. Why, if you knock out the cytokine, two of the chains of the receptor, you didn't have any significant effect, but now when you knocked out the gamma chain, now you had severe combined immunodeficiency. Any suggestions why that should be?
Isn't it that you don't need the alpha chain?
Well,
it can be low levels of IL-2 stimulation, which is gamma and beta chain.
Right, but if you knocked out interleukin-2, you had no interleukin-2 at all in the body, you also had minimal impact on the immune system.
Gamma chain is common to other receptors.
Exactly. You got to think a little bit out of the box. So it turns out that whereas the alpha and beta chains are specific for the cytokines, so for IL-2, IL-4, IL-21, IL-7, IL-9, and IL-15, they have unique chains that specifically bind to that particular cytokine. It turns out that the gamma chain is a basically all-purpose utility signal transduction receptor. And in essence, whereas these chains give specificity for binding of the cytokine, the gamma chain is what actually does the signal transduction. And in fact, if you knock out the gamma chain now, you not only don't have interleukin-2, you don't have IL-4, IL-21, IL-7, IL-9, and IL-15 function, and now it makes a lot of sense why now you have severe combined immunodeficiency, because each one of these cytokines plays critical roles, as shown here, in different stages of T cell and B cell maturation, and now you've knocked out a whole large number of cytokines critical for being T cell maturation. And the gamma chain is a unique molecule to immune maturation. And that explained why David's manifestations solely were his immune system, because the gamma chain really doesn't play a role in other aspects of physiology. It's not important in the liver and the kidney, et cetera, et cetera. Okay, is that clear? Now, if you then go into a immunodeficiency clinic and ask the question, what fraction of these patients have mutations? Half of them have mutations of the common gamma chain. A very small amount interleukin 7, and I'll discuss in a minute JAK3 deficiency, and I'll also discuss in a minute adenosine deaminase deficiency, which also can cause immunodeficiencies. Now, again, severe combined deficiency is the presentation, so David had knockout of his common gamma chain, but there's also a population of patients that the etiology is adenosine deaminase deficiency. And what's interesting about these patients is they can actually be relatively normal for a year or two after birth, but then they start losing their B cells and T cells. So what that suggested was that the problem wasn't an underlying maturational problem, but there was some toxin or toxic metabolite that was being generated as they were growing that was slowly killing off their B cells and T cells. And then it was identified the gene they were lacking was adenosine deaminase, and in fact, this is just a metabolic pathway. Adenosine, in order to eliminate it, it ultimately has to get deaminated and ultimately becomes uric acid where it gets secreted in the urine. If you lack adenosine deaminase, this is blocked, and you basically are going to get buildup of adenosine. And again, this isn't a biochemistry lecture, so you don't have to go into detail, but it turns out that the major problem, whereas adenosine has alternate pathway for being metabolized by phosphorylating it and then using an enzyme called adenosine monophosphate, deoxyadenosine lacks that, and therefore you get buildup of deoxyadenosine. Well, why should buildup of deoxyadenosine be toxic? And it turns out that deoxyadenosine is, as you all know, is the source of DATP. And DATP is a critical nucleotide that you utilize for making DNA. Well, in order to, and what makes it unique for lymphocytes is that lymphocytes, once they phosphorylate DATP, they have very, very low levels of the enzyme 5' nucleotidase to dephosphorylate it. And phosphorylated DATP can't leave the cell. So you end up doing is you're taking all your deoxyadenosine, you phosphorylate it, it gets built up, but you can't dephosphorylate it. So lymphocytes uniquely have buildup of this DATP, and that's why it's uniquely toxic to lymphoid lineage cells. Other cells are able to dephosphorylate it. Dephosphorylated deoxyadenosine could just diffuse out of the cell. So it's kind of like it becomes the sink for DATP. Well, why is that toxic? Again, one of the major reasons why it's toxic is that there's an enzyme called ribonucleotide reductase, and ribonucleotide reductase is a critical enzyme for generating all these deoxynucleotides for making DNA. However, enzymes, as you know, have feedback loops that tell it when it could stop because you have enough of the substrate. And it turns out that ribonucleotide reductase, even though it's involved in making dCTP, dTTP, and dGTP, the only feedback it utilizes is the levels of DATP. So if ribonucleotide reductase sees high levels of DATP, it basically says, I don't need to phosphorylate dGTP, dTTP, and dCTP. So now what's going to happen to your ability to make DNA? You can't make it because you lack the building blocks. And therefore and lymphocytes, as you know, have to proliferate. Well, if you can't make the nucleotides for DNA, you can't proliferate, and then it's ultimately toxic. And that explains why it takes a while for these patients to get immunodeficiencies, because they undergo proliferation, and then the cells basically make too many mutations because they lack appropriate DNTPs. Okay, another mechanism for severe combined immunodeficiency is Jakstat defect. If you recall, Jakstats play a role in what kind of signal transduction? Cytokines, exactly. So now, the same way if you knock out the common gamma chain and you lose a panel of cytokines, what happens if you particularly knock out a specific Jakstat that's critical for cytokines required? And again, this is just to review Jakstat. Basically, cytokines, Jak comes together, phosphorylates stat, and then the heterodimer gets into the nucleus and turns on genes. And it turns out now, if you look at the different cytokines, all these cytokines, IL-15, IL-4, 9, 7, and 2, all use JAK3. So now you can imagine if you knock out JAK3, what's going to happen to the function of these receptors. You're going to lose them. And then you basically have inability of maturation that's dependent upon these multiple cytokines, and it turns out you get severe combined immunodeficiency. So now, in 1990, the first gene therapy was performed, and it was performed for a patient that had adenosine deaminase deficiency. And again, to do gene therapy, you want to do it in a situation where you've identified the gene, and the level of the gene that needs to be expressed in order for normal cellular function, you should have a lot of leeway. Because one of the biggest problems with doing gene therapy is putting it in a way that the gene is appropriately expressed the way it is physiologically. But if you have a gene that, if it's twofold, threefold, 0.5-fold, it's good enough, then gene therapy is a lot easier to do. Well, adenosine deaminase deficiency, it was toxic metabolite buildup, so you just need a little adenosine deaminase, and that was able to resolve it. That's one of the reasons why this was chosen. And in fact, in this case, they actually did not treat hematopoietic stem cells. They harvested peripheral blood from a patient. They treated the peripheral T cells and B cells and gave it back to the patient. The reason for doing that is, first of all, it's a lot easier to do. Second of all, you don't have to worry about introducing mutations into a hematopoietic stem cell that can then go on and develop malignancy. And also that adenosine deaminase patients, they had mature T cells and B cells, just if you let them go too long, they would start deteriorating. And these patients also can be treated with enzyme replacements. So they also can be treated by giving them pegylated adenosine deaminase, and so these patients had a reasonable level of B cells and T cells. And it turned out that of 2 patients treated, peripheral blood T cell counts in patient 1 increased and became normal, and patient 2 showed an increase. So it looked very, very promising that it was working. In 2002 and 2003, a much more aggressive gene therapy approach was utilized. Basically, these are children that had, what David had, gamma chain deficiency, severe combined immunodeficiency for that reason. And these patients, you had to treat their hematopoietic stem cells because you couldn't, they really didn't have a lot of mature B cells and T cells to treat. And in this case, in this group in France, 9 of 11 children were successfully treated with hematopoietic stem therapy, which is a mind-boggling thing. I mean, you had a previously untreatable congenital immunodeficiency, and now by gene therapy you could basically have a child that could leave the bubble and have a relatively normal life. Unfortunately, 4, I think it may even be 5 now, of these children developed leukemia due to insertional mutagenesis. So the retroviral vector they utilized inserted adjacent to a oncogene, and that caused malignant transformation. So that really has cast a pall upon the field of gene therapy for using hematopoietic stem cells. But an additional issue, particularly for this kind of stem cell therapy, is that you're giving a growth factor receptor. And you can appreciate if you express too much of a growth factor receptor, that's also going to make cells proliferate too much. And the combination of mutation plus a hyperproliferating cell is a recipe for potentially malignant transformation. If you're doing gene therapy with a gene that is not associated with proliferation, you may not have as high an incidence. Now, if we move down the road to DiGeorge syndrome, it's decreased T cells, normal B cells, decreased IgG, and the patients present with hypocalcemia and seizures. Now, you're probably thinking, this is an immunodeficiency. Why should it be presenting with hypocalcemia and seizures? So we'll get to that in 1 second. Because now, the other point I want to make is that even though they have normal B cells, if you lack T cells, what impact is that going to have on your B cell function? You're right, you won't make IgG, you won't make IgA, only IgM. So even though you have functional B cells, it's like having a soccer player and you tie both hands, well, in soccer, if you tie both hands behind the back, it doesn't really matter. It just means they won't get a penalty. But if you kind of like put a 50-pound weight on his or her leg, it's going to compromise them. So even though the body is there, they're not as functional. So therefore, they have functional immunodeficiency based on their lack of IgG, so they get bacterial infections as well. And the etiology of it is a deletion in chromosome 22q11. Therefore, it impacts on maturation, mediastinal maturation, and the third and fourth brachial pouches do not develop normally. And again, the treatment potentially for this patient can be thymic transplant. Now, why do these patients have hypercalcemia and seizures? And how could you make this diagnosis using an X-ray? Well, the tissue that these patients lack is the thymus. And you could actually do a lateral chest X-ray, and this is of a baby, and here is the heart, and right here in the anterior mediastinum is the thymus. That's a perfectly normal child with a normal thymus. If you look at a patient with DiGeorge syndrome, and now you look at their cardiac, do you see something? In an X-ray, black means nothing's there. So you see there's nothing here. They have a heart, and what do you think is lacking? The thymus. So you could basically make the diagnosis without even being an immunologist, just doing a lateral chest X-ray. Well, why do these patients have hypercalcemia? Is because in addition to the thymus, the lack of the third and fourth brachial pouches also means they lack a parathyroid gland. And the parathyroid gland plays, hypocalcemia. The hyperparathyroid gland plays a critical role in controlling calcium metabolism. So they're presenting with seizures and hypocalcemia because they lack a parathyroid, not because of a problem with their thymus. Now, I said in the beginning, how can knowledge of immunology prevent you from killing a patient? And the answer is, well, this child has no functioning T cells. He's a day or two old. You're doing all these blood tests on him, and then you finally, nurse tells you the patient is anemic because you've taken so much blood. And when a patient's anemic, what do you do how do you treat that? You give a blood transfusion. That makes a lot of sense, right? Well, if you give this patient a blood transfusion, what potential problem can this child have? Well, but that you could choose. What immunological problem could this child have? Well, a transfusion reaction, but be more specific. What unique transfusion reaction would a child that lacks T cells have? And what I'll say is that in the red blood cell transfusion, there are a few T cells that are mixed in that will also be given to the patient. Graft versus host, because these T cells get inside the patient, and they look around and they say, whoa, everything's foreign here. We've been invaded. The entire body's been taken over by foreign MHC molecules. And now what they do is they now start proliferating out the wazoo, and they start attacking the recipient because they think they're doing a good job. They're protecting themselves from invasion. And the reality is they're completely messed up. They're the invaders. So therefore, if you would give this child a blood transfusion, this child would have horrible graft versus host disease eventually, and you can't reverse it. You can't take those cells back out. So once you've made that mistake, ultimately you could lead to the death of this child. So therefore, but how would you treat blood to prevent those lymphocytes from proliferating? How do you treat cells to prevent them? You irradiate them. For red blood cells, irradiation is no problem because there's no nucleus in red blood cells. But if you irradiate the blood, now those few T cells, you cross-link their DNA, they get into the body, they say, oh, we got to proliferate to fight this foreign cells, but they can't because you've basically, again, you've castrated them, they can't replicate, and therefore they can't do any harm. So therefore, if patient in the first day or two of life has hypocalcemia and seizures, do the lateral chest X-ray, you see there's no thymus, that tells you do not give this baby a red cell transfusion unless you've irradiated the blood products, and that way you won't kill this child. Okay? Question?
If a child had T cells, how would that stop graft versus host?
Okay, so if a child had T cells, how would that stop a graft versus host? Because all of us all of us don't get but we frequently transfuse patients and they don't get graft versus host. Well, let's think about it. You have a war, and on one side are 100 soldiers, and on the other side are 1 billion soldiers. Who's going to win?
Can T cells fight T cells?
Well, the same way that the T cells that come into the body see the body as foreign, our T cells would see the transfused T cells as foreign and eliminate them. So routinely, and again, some patients do get transfusion reactions for that and other reasons. But at the end of the day, if you have normal T cell function, no big deal to eliminate that handful of T cells. It's only if you lack T cells that would be a problem. Okay? And this is just showing that they actually have an interesting looking facies because it's a whole midline maturational problem. Okay, so now, and this is just to kind of reinforce the point that if you lack T cells, you actually compromise B cell function. And again, as I had mentioned, helper T cells require a second signal provided by interaction of CD40 ligand expressed by helper T cells and CD40 that's present on the surface of B cells. And now, if someone has a mutation in their CD40 ligand, what kind of antibody deficiency do you think they would have? They would lack IgG, lack IgA, and in fact, I'll discuss that in a minute. It's called hyper-IgM syndrome. But again, lacking T cells, these patients would only make IgM for the most part. Okay, now we get into X-linked, now we're getting into immunoglobulins. Common one, X-linked agammaglobulinemia, decreased B cells. They lack antibody completely. They're going to present after 6 months because of maternal antibody. They're infected with bacteria. And in this case, their defect is BTK, which I had mentioned briefly, is a critical enzyme required for B cell maturation. So if you lack this enzyme, you specifically don't mature B cells, and then you don't have any antibodies. The treatment for these patients, you just give them IV gamma globulin. So the same way that maternal antibody protects the baby first to 6 months, if you give these children monthly gamma globulin injections, they actually do quite well, but they need to get this continuous treatment for pretty much the rest of their lives. But as opposed to a T cell defect, you can't transfuse T cells to these patients, so that makes immunoglobulin deficiency a little easier to treat. Now, hyper-IgM syndrome, I just mentioned a second, they present after 6 months. And one possible etiology would be CD40 ligand deficiency. Can anyone think of another critical enzyme in class switching that could also cause you not to be able to make IgG or IgA? What? AID, exactly. AID is critical for class switching. If you don't have AID, you can't switch. And in fact, one causes the absence of CD40 ligand, but as you so nicely remembered, the other one is lack of AID. You can't basically recruit all those repair enzymes to cause class switching. And again, what I want to underline to you is that this really allows us to appreciate that these enzymes do what we say they do, and they're the only ones doing it. Because if you knock that out, there's nothing to pick up the slack. So again, that's, as I started the talk by saying, that's why these patients are so instructive in teaching us how the immune system works. Because now I can tell you that you could take it to the bank that CD40 ligand is absolutely important for class switching. I could tell you, take it to the bank, AID is absolutely important in class switching, because if we have individuals that lack that, they don't have the class switching. Okay? Okay, and this is just showing, again, the CD40 ligand. Okay, now there's also subclass deficiencies, and they basically have deficiencies of subclasses, and the major etiology is unknown, but there's been reports of just, if you have mutations in the switch regions of the immunoglobulin molecule, you can appreciate you're not going to be able to bring them together to loop out the appropriate ones. Now, IgA deficiency, I mentioned yesterday, decreased IgA, bacterial, and failure of isotope switching, but the question is that they really don't have dramatic deficiencies, and now you know why. Because what's picking up the slack for these patients in their gut? No, what humoral factor can also IgM, because IgM could also bind to the has a J chain, and it can also be transported across epithelial cells and could pick up some of the slack. Their lymphocyte syndrome is quite an unusual immunodeficiency, but these patients have markedly decreased T cells and they have normal B cells. And it turns out that, as the name implies, they're bare because they lack MHC class II. It's a mutation in the MHC class II promoter. And therefore, now if you don't have MHC class II, what kind of T cells won't be able to mature in the thymus because they won't be able to get positively selected? CD4. So these patients really lack CD4. So it's, again, clinically like severe AIDS because they don't have CD4 cells. And in addition, if a macrophage is infected, it doesn't have a class II MHC molecule, even if it did have some CD4 cells to present to. So therefore, these patients have very significant deficiencies. And again, they can be treated with bone marrow transplant. And this is just a slide to show you all the different genes in the context of activated T cells and B cells, and again, showing you how all of them are associated with immunodeficiencies variable. So now, the only other one I want to mention is TAP deficiency. So if you recall, what does TAP do? I'll give you, it's a pump. What's it pumping?
MHC 1 and 2.
Okay.
It allows the...
So MHC class 2 is getting loaded in the vacuole, so it's not going to need to get pumped peptides. So what's going to need to get peptides pumped to it? What MHC molecule needs to get loaded with peptides in order to leave the endoplasmic reticulum? MHC class 1, and TAP actually does the pumping. So it turns out if you don't have TAP, you can't load the MHC class 1 with peptides, and then you can't express class 1 MHC. Now you don't express class 1 MHC, what kind of T cells won't you see in the periphery? CD8. So these patients actually have deficiency in CD8, and they tend to get a lot of viral infections. Okay, so Job syndrome is actually, the English are always a lot more interesting than the Americans, because, and now I get Veronica's attention here. I mean, Victoria's attention. So the reason is that when Americans discover something, they name it after themselves. Because, you know, America is a relatively egotistical country. Right? I'm sure you all know that by now. In England, though, they tend to be a lot more poetic. They have a classical education. So if they see something interesting, they try to relate the discovery to something in the arts and sciences or literature. So who is Job? People know who Job was? Who's in the Bible? And what disease or problem did Job have? He had skin boils. So these children presented with cold abscesses, and the individuals looking at them said, oh my gosh, you know, this kid looked like Job. And then it was called Job syndrome. You know, if it was discovered in America, they would have named it after whoever discovered it. These patients also have very high levels of IgE. The problem is that the white blood cells can't get to areas of infection. They have cold abscesses. Normally, if you have an abscess, it's hot because you have the erythema, you have the vasodilation. Their abscesses are cold because the phagocytes just don't get to where the infection is, and they can't release tumor necrosis factor, all the things that are required for the vasodilation and erythema that gives you the warmth. They get infected with staph, strep, and candida. And it turns out that we now know that the etiology is a mutation in STAT3. So, and again, STAT3 plays a role in the signal transduction required for chemotaxis. And the treatment, these patients are on chronic antibiotics. In contrast to Job syndrome, another phagocytic deficiency is chronic granulomatous disease. And these patients also have ineffective phagocytosis, but what they're characterized, they have hot abscesses. And the reason they have hot abscesses is because the phagocytes can get there, they can secrete their factors, they just can't kill the bacteria. So therefore, that's why the infection can't be clear, but the abscesses are hot. So that's great, because you have a patient that has a lot of abscesses, you put your hand on the abscess, and if it's cold, Job syndrome. If it's hot, chronic granulomatous disease. Now, obviously, you do the lab test to confirm it, but it makes you feel good that you can just kind of lay your hands on a patient and quickly make the diagnosis. And again, the defect is due to the NADPH oxidase that's required in order to get this oxidative burst to kill bacteria inside the vacuole, the phagolysosome. And again, treatment antibiotics, gamma interferon seems to increase, as you know, the phagocytic capacity of macrophages, and therefore you could generate enzymes that pick up the slack and allow it to kill better. Okay, so now to summarize and put things into context, so if you look at the maturational pathway, defects at different stages have impact downstream. So if we look at B cell and T cell maturation, if you have a mutation, for example, that impacts on the capacity of stem cells to become lymphoid progenitors, in this case, adenosine deaminase deficiency, gamma chain deficiency, then you basically lose B cell and T cell maturation. If you have a mutation in another gene, for example, RAG, which is required for B cell gene rearrangement and T cell gene rearrangement, again, you lack B cells and T cells. BTK is only required for B cell maturation, so if you knock this out, you basically have no B cells, but you have normal T cells. In contrast, if you have lack of thymus, which is required for T cells, you lack T cells but have normal numbers of B cells. Does that make sense? Right? And now, if you have either a defect in class 2 MHC, then will have defect in maturation of CD4 T cells. If you have a mutation in TAP1 or TAP2, then you'll have a deficiency in CD8 T cells, right? And now if we look at fine-tuning in terms of the differentiation of B cells and T cells, clearly there's a whole host of defects in T cell signal transduction molecule, so you'll have normal numbers of T cells, but they won't be able to be activated and proliferate appropriately. If you have a mutation in CD40 ligand, you can't class switch, so you have hyper-IgM syndrome, and it's not shown in this slide, but also AID, which I'm so proud that you remembered, also would have hyper-IgM syndrome. And again, there's a whole host of switch factor mutations in the immunoglobulin genes that give you IgG subclass immunodeficiencies. Okay, so basically, patients provide many insights into working of the immune system because a lot of the genes that I've mentioned play a critical role in differentiation actually turn out to be the case because if patients lack those genes, they manifest the inability to undergo that maturational stage. Clinical history helps diagnose the etiology in terms of age of presentation, what pathogens they present with, and ignorance of immunology can kill your patients. And an example of that, for example, is a patient with hypocalcemia and seizures. You want to know that they have DiGeorge syndrome, do a lateral chest x-ray, because if you would give them a blood transfusion, they would develop graft versus host disease. Okay, thank you very much for your attention. And now we have one more lecture to go and we're done. Thanks a lot.